So, class, let's focus on some of the phenotypic techniques we have. Our phenotypic methods are going to focus on what we can observe with our eyes, either with a microscope or with our gross anatomy, with the gross eye itself, an unmagnified method. Our common phenotypic methods are typically going to be used for presumptive diagnosis. Phenotypic methods are no longer the gold standard. The gold standard is our genotypic methods. But if we need quick data to make a quick diagnosis or a quick preliminary diagnosis, phenotypic methods are cheap and they're reliable and they're fast. The two most common stains we use for our phenotypic methods are that classic Gram stain and the acid-fast stain as well. When we are cultivating our specimen, we need to isolate pure cultures to get our good samples so we can have good data. So we'll use specialized media. Some media is going to preferentially select for one kind of pathogen and destroy the other kinds of pathogens. This specialized media is sometimes going to be formulated in such a way that pathogens that normally wouldn't be able to grow in regular generic media like nutrient agar would now be able to grow in very high concentrations. So we could have specialized media that instead of containing glucose as a carbon source is going to contain galactose as a carbon source. Our selective media is when we are working with non-sterile specimens. This selective media will allow one kind of microorganism to grow and another kind to not grow. Classic examples of selective media include EMB agar or mulin-hindrin agar, or we could have our triple sugar iron agar as well. These media are going to allow some things to grow and other things not to grow. We could also use differential media. Differential media, generally speaking, allows pretty much everything to grow, but depending on what grows, it's going to have some kind of different fermentation, some kind of different color change occur. Many media types are both differential and selective at the same time. So that media can select for only gram-negative organisms, and depending on what kind of gram-negative organism, it will turn green, or will turn yellow, or remain pink, depending on what kind of pH indicators you put in the media. Our pure cultures, generally speaking, are going to need to be obtained and isolated from the selective medias, and those pure cultures can then be used for additional tests to help get a more accurate and precise diagnosis. Physiological reactions of the bacterium to the nutrients the bacterium are trying to be grown in are classic methods for identifying our bacteriums. Some bacterium are going to behave differently in the presence of oxygen compared to others, the presence of carbon dioxide, the presence of methane gas, the presence of sulfur-containing compounds, all can be used to look at different kinds of bacteria and how they physiologically respond to these different situations. Many metabolic reactions or chemical reactions in the bacterium are controlled by enzymes. We spent a lot of time earlier in this course talking about how enzymes control chemical reactions, and these metabolic reactions that are controlled by enzymes can be visualized with the color change. So if we culture our microbe in a special medium with a special substrate, we can then test for a particular end product. Sometimes, if this particular end product is an acid, maybe if we're trying to put the bacterium in a situation that it's going to cause fermentation to occur, if that bacterium is capable of fermenting the carbon source, it will likely excrete an acid. That acid can then change the color of a pH indicator, and our media will then go from red to yellow, or it can go from pink to clear. No coloration means that if there's no coloration or no change in color, many times that means that that bacterium lacks the enzyme that's capable of utilizing that substrate. So if we take that bacterium and it is capable of growing on the agar, but it doesn't have the accompanying color change, that means that special ingredient we added can't be used by the bacterium, and that's an important diagnostic technique. Whether or not that bacterium can ferment lactose, for example, is a common diagnostic technique. So we can have our mystery microbe, and we put it on some special media, some differential media. If the enzyme is present in the microbe, that enzyme is going to cause our substance that we put in the media to be changed form a product, and that product generally is going to give us a color change. Woohoo, positive result. If that enzyme is absent, then there would be no product form, and then there would be no color change. So we'd have a negative result. Many of these tests are going to have known positives and known negatives performed side by side with your unknown culture. So you have some frame of reference to compare your results to from your experimental sample. So again, a very prominent test that we can look. I've mentioned repeatedly carbohydrate fermentation and the production of an acid end product. We also could have the presence of catalase in our coagulase in our bacterium that can hydrolyze gelatin, or if we could have other enzymes that are present to hydrolyze other specific polymers. We could also have enzymes that are related to the presence of oxygen, so we could have catalase, oxidase, or our coagulase. Various byproducts of our metabolism could include some of the miscellaneous amino acids that are produced, or whether or not there could be a specific protein that contains high concentrations of known metals. Sometimes we can use rapid miniaturized systems to perform many tests at the same time. In labs that have lots of money and where the budget really isn't a problem, these rapid tests are used frequently. And they can perform up to 23 different tests at the same time. Here's a classic example of a rapid test. During this rapid test, you will take a sample of your bacterium on a stick, and then you'll use that stick to puncture each and every single one of these wells. And then based on the color change that occurs in each and every one of those wells, you can say negative result, negative, positive, negative, positive, negative, 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 positive. And then you can use a numbering sequence or cheat sheet from your wells to then add up your scores and get a numerical code that then tells you what your bacterial species is. These are almost too easy. Sometimes we use dichotomous keys. At an undergraduate level, having students collect lots of data, create a dichotomous key, and then use their dichotomous key to determine the composition, the species composition of an unknown sample is a very classic lab technique. Our dichotomous keys are going to be based on very easy to perform tests with very clear-cut results. Gram stains are on all dichotomous keys, and we'll also typically look at the metabolic reactions to oxygen, motility, or some other quickly performed phenotypic tests. These tests are then used to make a flowchart, and eventually at the end of our flowchart, we'll have the name of a specific microorganism. So we can start off with just a simple test. Look on the light microscope. Tell me what is the shape of the microorganism. Is it a spherical cocci? Is it a comma-shaped bacilli? Or is it a squiggly spirorette? And then you can say, is it gram negative, gram positive? Let's say arbitrarily it was gram positive. And then after we determine it's gram positive, we could perform the catalase test. And oh my goodness gracious, the catalase test is positive. So now that we've had a positive catalase test, we're going to need to perform 2 more tests to determine its oxygen use. And if it's strictly aerobic, we can know that it's Micrococcus. If it's a facultative anaerobic, we can know it's a Staphylococcus or a Planococcus. Good dichotomous keys always end with one species at the end. This is a bad dichotomous key. It's a bad dichotomous key because right here we have 3 species, right here we have 2 species. So if you ended up with this result, you'd still have to do more tests to determine which of those 3 bacterial species you were working with. Another less common test is phage typing. Recall, a bacteriophage is a virus that infects a bacterium. We typically will use bacteriophage or phage typing when our normal phenotypic methods aren't up to the task of differentiating our bacterium. So we'll start off by growing a very carpet of bacterium on our Petri plate. And then we'll divide some sections using a permanent marker and draw them out onto the Petri plate. And in each section, we inoculate it with a different kind of bacteriophage. Our bacteriophages are very specific. They will only infect a specific strain of a specific species of bacteria. And if we see the carpet of bacterium cleared in that section of the plate, then we know that that bacteriophage was able to grow and reproduce, and it was capable of infecting the bacterium. So here's a classic bacteriophage plate. Notice how it's broken into the segments, and it was completely covered in bacteria, but in zone 52A and zone 79, the bacteria was killed off by the virus, which means that whatever that specific bacterium is, it can be infected by that specific bacteriophage. So now we can deduce what kind of bacteria it was based off the fact that the virus was growing on it. Some other miscellaneous tests include animal cultures. When working with Mycobacterium leprae, the pathogen that causes leprosy, or Treponema pallidum, we need to culture that bacterium in an animal host, in vivo, in a living organism, to determine an accurate diagnosis. Many times that living organism is going to be an avian embryo. Sometimes, though, we're going to need to use an antimicrobial susceptibility test. This is known as the Kirby-Bauer assay, or as I like to call it, the zone of inhibition test, where we are going to plate out the bacterium and then drop little discs containing antibiotics onto the plate to determine what drug the antibiotic is resistant to. Another important thing we need to emphasize when determining our culture species, or what species of bacteria we're working with, is whether or not what we're testing was from the sample, was it a pathogen, or whether or not it was part of the normal microbiota. Our bodies are crawling with E. coli. We have E. coli on the surface of our bodies, but not much. But if we look in our bloodstream, we should have almost no E. coli. And if we look in our colons, we should have tons and tons of E. coli. So if we look at a urine sample and there's just a little bit of E. coli in it, not a big deal, because E. coli is all over the place. Now, if there was a high concentration of colony-forming units of E. coli, then that would be a big deal. That would be indicative and say, okay, that's probably not contamination of normal microbiota, but instead, that's probably some pathogen that we've isolated from the sample site. Now, if we're looking at other species that are known to only be pathogenic and not exist on us as a part of our normal microbiota, a single colony of those species, such as Mycobacterium tuberculosis, could be very indicative of a full-blown infection. So depending on the microbe species, we can maybe say, oh, okay, it's probably just a contaminant if it's at a low level and not a high level. But depending on how pathogenic the species is, even low levels can be determined to be the source of infection. One way that we can help minimize misdiagnoses from our contamination from resident microbiota is to have repeated samples collected of the same fluid from different points in the person's body. If you're having a patient that's getting their blood drawn to get blood work done for microbial species, that blood needs to be drawn multiple times from multiple sites on the body. Maybe, just maybe, there happened to be a pathogen at the first pinprick, or where the first needle went into the patient, and that pathogen or that microbe contaminated the first sample, but the second sample didn't have that microbe present to contaminate it. Now, if we can get repeated isolation of the same microorganism from the same fluid collected at different points or different ways from the patient, that's probably going to be a big red flag that that is going to be not a normal microbiota, but part of the disease-causing agent. So, concept check class. You've just obtained a fecal sample from a patient. What type of growth media would you use to grow a suspected pathogen from our fecal sample? Would you use a differential media? Would you use a carbohydrate fermentation media? Would you use a selective media? Or would you use a media containing an antibiotic? Or all of the above are correct. If you don't know the answer, that is okay. Rewind the video. Here's a big clue for you. We have many different species of bacteria present in our feces. I'll say that again. Our feces are a chemical or a cocktail of bacteria and viruses. So what would you do to isolate our pathogen from the sample? Pause the video, rewind it, check your PowerPoints, check your textbook, and get me an answer. 1, 2, 3, 4, 5. The correct answer is E. All the choices are correct. Because our feces are such a cocktail of different microbes, you would want to use the full battery of testing techniques to help differentiate what is and what is not pathogenic from the feces. If you have any questions about this presentation, please feel free to post them in the discussion board or shoot me an email. Happy studies.